Hello, everyone, and welcome to this life science event here from the University of Oslo. My name is Alexander Jensenius. I'm a professor of music technology here, and I'm also a member of the Abino project. And today we had originally planned that we should meet at the Litteraturhuset downtown in Oslo and have more of a physical event. As it happens, we are not doing that. We are mainly doing then this as an online event. And also for Corona measures, we have decided to pre-record some of the introduction lectures. So we're going to watch them now very soon. And then afterwards, here with my colleagues from the Abino project, we will have a panel discussion. And then as you're watching this, feel free to also use the chat here on the side of the video that you're watching. Please ask questions there or add comments, and we will pick up on those later in the panel discussion. But then I would like to give the word to Hanne Scholz, the leader of the Abino project, to present today's program.
Welcome to this event at Oslo Life Science Conference 2021 with the title Music for Stem Cells, Aiming to Cure Diabetes with Sound Waves. I will start to introduce you to the Abino project. I'm the leader of the project, and the Abino project is one of the conversion environment at Oslo Life Science. The conversion environment are united interdisciplinary research group that address major challenges in health and environments. We want to develop novel and future diabetes model by introducing new strategy for stem cell differentiation. How shall we do this? We have assembling the best researcher from very interdisciplinary field at University in Oslo. Our expert from the Rhythmo combines methods from musicology, physiology and informatics to study the rhythm as a fundamental property of human life. Our expert from physics department are Norway's most comprehensive researcher, and they also host the Center of Excellence for Education. Our experts from the Center of Hybrid Technology Hub, Organ-on-a-Chip Technology, aims to develop organ-on-a-chip platform that can reproduce the core functionality of the human body. Why diabetes? Our blood sugar is regulated by the islets in the pancreas. When the blood sugar is high, the islets secrete insulin. And insulin opens up for glucose from all cells in our body, so glucose can enter in and be used as energy. If the blood sugar is too low, the alpha cells in the islets producing glucagon, and then help to increase the blood sugar. And this very, very fine-tuned regulation are going 24/7 in repeated cycling. Importantly, too high blood sugar and too low blood sugar is very detrimental for our body and damaging the organ and can also be deadly. So, when you are diagnosed as a type 1 diabetic patient, the beta cells in the islets are destroyed by immune cells, so it doesn't produce insulin. And without insulin, there is no blood sugar regulation. So the body needs insulin to regulate the blood sugar. Until the discovery of insulin more than 100 years ago, all patients that suffering from type 1 diabetes died within 1 to 2 years. So then, now we have insulin and patients are lifelong dependent of insulin. However, many patients struggling with management of the disease. Constantly blood sugar measurements and insulin injection are difficult. Too much insulin, too little insulin is both detrimental and can be dangerous. This also leads to fluctuation of the blood sugar. So even though you have insulin, the blood sugar is high and low and high and low during the day. These fluctuations of blood sugar are leading to complication because it's detrimental to the organ. And this secondary complication can lead to heart attack, kidney failure, blindness, stroke, amputation. So insulin are treating the symptoms of the patient, but there are no cure yet. The hope and the expectation lies in replacing the insulin-producing beta cells with healthy cells. How can we do this? It's really a pleasure for me to give the stage to our excellent research fellows at the Albino Project with expertise in biology, physics, and musicology to show you how they work, think, and connect with each other to solve this very difficult task. Enjoy. Thank you.
Hi, this is Chen Chengwang, a PhD student at University of Oslo. And today I'm going to talk about diabetes and how could stem cells help. So in 2016, the US FDA approved the first insulin pump that can automatically monitor the glucose, supplying insulin when the blood sugar are high and cut off the insulin supply when the sugar levels start to drop. When I was talking about my research with a student at the University of Oslo, I was being asked a question. You say the automatic insulin pump works quite well, why we have to still working on making beta cells. I was surprised and delighted about her knowledge was updated with all those exciting achievements. And then I start to tell why we still need research on beta cell regeneration. It is my great honor and pleasure to share the idea with you today. So this is a recently launched movie named Greenland, and the family will be transferred to a safe place and trying to escape a disaster. And when the plane was just going to leave, and they found that they forgot their kids' insulin. And it's quite reasonable that they might forgot something somewhere. However, type 1 diabetes cannot survive long without insulin supply. And you can imagine that it will be never easy to enjoy a really normal life. So again, this graph published 2 months ago shows that the average life lost in type 1 diabetes patients is 7.8 years long. So it's quite a lot. So, by the way, if the audience unfortunately have diabetes, the situation will be better if we have a good daily glucose management. So the best glucose sensor on the market tells the glucose level every 5 minutes. However, the beta cells in our body sensor the blood glucose level hundreds of times in a second. Instead of daily medication, worrying, pain, why we do not just replace the lost beta cells in our body with the healthy cells, or just let them come back if we can. So I have to mention that we are not talking about inject antibiotics to cure coronavirus. So never inject an approved cell product into your body without a professional doctor's guide. So actually, it was not a new story or a crazy imagination. Back in 1938, a book was published named The Culture of Organs. To some extent, humans actually have the capacity for regeneration. For instance, specific cells such as bone marrows can reproduce blood cells. And when a small piece of liver has been removed, it can self-renew. However, humans cannot perform the same tricky of regrowing limbs as salamanders can. And unfortunately, the islets cannot too. This is because during the embryonic development, most of the mammal cells lost their pluripotent potential. So games changed because human embryonic cells were found in 1998. They are the cells isolated in the blastula, a very early stage of human embryo. One embryonic cell can self-replicate into billions of cells in a petri dish, and they have the potential to be any kind of cells in the human body. However, because of the ethical concern, generating new embryonic cells nowadays are strictly forbidden in many countries, including in Norway. 9 years later, a great scientist, Yamalanka and John, find a way to reprogram the adult cells into pluripotent stem cells, and they are similar with embryonic stem cells. Nowadays, even fat cells can be reprogrammed as stem cells. You know, it's how easy to get fat cells these days. And this achievement really boosts the research of stem cell-based therapy studies. But how to generate beta cells from iPS cells is another challenge. To achieve this, we have to learn from the body to speak the language that the cells have, to tell them, let them to be the cells we needed. So by adding chemicals, proteins, growth factors, and build up the environment the cells like, and inducing different genes expressions sequentially, like this slide shows, step by step, we can eventually get insulin-produced cells within 30 days in a petri dish. So this slide on the left side shows how the islets grows in our lab. And the video on the right side shows how the insulin-producing cells react with high concentration of glucose. In moment means the cells are taking up the calcium and pushing out the insulin. So in 2002, one of the most famous scientific journal, Science, on their cover page published how people's imagination at that time the bionic human will be like in the future. That is the beginning of regenerative medicine. As science moving forward, ideas changes now. This is expectation in 2016 of how regenerative medicine looks like in the future. And we will replace the unhealthy part in our body with a more natural solution. So the gift of insulin and insulin pump transformed the deadly disease into a chronic diseases, which requires intensive daily management. Through changing the fuel islets, patients do not need to carry a pump every day while skiing, camping, or swimming. And they do not need to do extra exercise as you and I did every day. And they do not need to worry about diet and blood glucose every now and then. So achievement are continually making from scientists around the world and also in our lab. But methods to making beta cells from iPS cells are still not perfect enough. It's expensive, complex, and time-consuming. They have realized nowadays that it would be really interdisciplinary research to fill the gap. Luckily, experts from almost every field are gathering together to make this happen. Well, as a stem cell biologist, I'm really proud to work with Dr. Hanna and have a chance to update my knowledge by working with experts from different fields. Try to contribute to this no matter how big or small it is. In the future, picking islets for diabetes treatment will be just like picking apple, and I wish it will be a lot later than Elon Musk moving to the Mars. Thank you.
Hi everyone, my name is Thomas Combria and I'm working on the Abino project and I'm part of the physics department. And today I will try to talk a bit about acoustics and biology in the Abino project. So to start simple, what is acoustics? So acoustics is basically the study of sound waves. And what is a sound wave? A sound wave is just a local change of pressure in time that does propagate in a medium. So to take an example, for instance, if you have a loudspeaker producing some music or some sound, this loudspeaker will actually produce a pressure wave which will leave from the loudspeaker toward a target, for instance, your ear or a microphone. As we can see on this animation, so this pressure is oscillating up and down, and that's what does carry the sound. And we also see that it needs a medium, to propagate. So it doesn't propagate in vacuum, but sound waves do propagate in air. That's what we usually use, to talk, but also in liquids or in solids. What is interesting is that if we zoom on this medium and we look at what is actually going down at the molecular level of, for instance, the air of the liquid in which a sound wave is propagating, we can see that actually the sound will make the molecules or particles composing the medium oscillate. So on this animation, which is basically the same as the previous one, I just added the molecule constituting the air, for instance. And you can see that actually at the top part of the wave, you have more particles because the pressure is higher, and that's basically the definition of pressure. But you can also see that even though it seems that all this high density bunch of particles are propagating alongside with the wave, actually they are just oscillating around an equilibrium position. So in order to see that more clearly, you can focus on the black particle, which is in the middle of the screen, which is just defined by its color. And you can see that it's actually oscillating back and forth, so that a sound wave is not propagating any matter. No matter gets displaced, they are just oscillating back and forth. So why is that interesting in terms of the Abino project? So now we can think it of in terms of biology. So if we input a sound wave, for instance, inside a body, or if a body is subjected to acoustic waves, this will actually make the component of the biological materials, cells, oscillate back and forth like this. And that will be principally the biggest effect on the principal effect that you can expect when inputting sound wave inside biology. Other things can also happen which are basically consequences of these oscillations. So when you input a sound wave inside a body, you can also expect some eating, for instance, of the cell. And that's actually used in medicine in order to remove tumors or cancer cells by just ablating them using heat to make them die. Other thing that can happen is that you can create bubbles when you put too much intensity of the sound. And that's actually what is happening, for instance, when you in decompression sickness, where you have bubbles just popping out in your bloodstream, and that can be very dangerous, for instance, when these bubbles are blocking bloodstreams, for instance. Another effect is that acoustic wave can bounce, and that's happening all the time when you are speaking on walls and things like that. But in the human body, they can also bounce on different kinds of tissue, for instance, on bones and on organs. And that's actually the principle which is used in ultrasound imaging. And there are a lot of other effects that I won't talk about today. So just to sum up, acoustics is just a way for us to input some mechanical energy inside a biological material. So as I already told you, acoustic waves on acoustic are used on a daily basis in hospitals in order to do, for instance, ultrasound imaging, which is kind of diagnostics, but also in order to cure people, for instance, to remove deadly cells such as tumor or cancer cells. On a more fundamental point of view, it is known in biology that cells do react to mechanical stimuli, which in a process called mechanosensing. It means that, for instance, if you probe a cell with a mechanical signal, if you push it, if you stretch it, if you compress it, then the cell will react and that can trigger some biological change at the cell level. So the main question I'm trying to address in this project is it possible to influence a cell behavior using acoustics as a mechanical stimuli? So just to follow up on what Chen just talked about, stem cells that we are now able to produce from whatever cell from your body can become any type of cells. And in particular, in the Albino project, our goal is to try to make stem cell into beta cells which are producing insulin. And the way this is usually done is to use drugs and chemicals in order to influence the cell differentiation in order to go toward the insulin-producing cell. But there are also new trends in biology trying to use other kind of stimuli, such as mechanical stimuli, in order to help or even to trigger this differentiation process. And that's why we are just trying to use acoustics in order to help stem cells to differentiate into insulin beta producing cells. So just to finish about what we are actually doing in the lab, and so what is my work on an everyday basis. So it's not clear for us as physicists to understand what is actually the effects of ultrasounds on cells, on biological material. So it has been claimed for years in ultrasound imaging that it's completely doesn't have any effects or bad or good effects. On the other hand, some people are using that in order to cure or to remove cells. So we know that something at some point should happen. So in order to understand what is happening at the biological level when using acoustics, we are, for instance, doing experiments where we can monitor cells undergoing acoustics signals. So, for instance, on the movie here, you can see a bunch of cancer cells actually undergoing an acoustic excitation. And if you focus on the two in the middle, you can see that they are actually undergoing an oscillation because of the acoustic wave. We can actually see that they are kind of dancing in the acoustic field. And so that's the first part as a physicist to understand what is going on, is just to monitor actually what are the direct effects, so the physical effect. And the second part in my job is to try to understand what these stimuli are actually doing at the biological level, so just trying to get a biological output out of this. So, as a physicist, we like to reduce things to a very small set of parameters. And for instance, we are now wondering what are the important parameters in the sound wave that can influence the cell behavior. And there are a few that you can think of straight out of the box. So for instance, one can think about the intensity. So here you have a loud sound, whereas here you have a very silent sound, which is that we can call the volume, basically, of the wave, if you speak in terms of music. Another thing that we can think of is what we call the frequency, or that musician calls the pitch. So you can have a treble, for instance, kind of sound, like this, or you can have a bass sound, like this. And that is another parameter that can or not change actually what is going on at the cell level. And the last one that we can think of, but I'm letting the next speaker talk about a bit more about that, is what we call the repetition rate. So basically, is the sound continuous, or is it segmented, or does it have a kind of a patterning? Like this. So basically, my whole job in the Arduino project is to broadcast music to cells in order to see if they actually react or not to this. Thank you very much.
Hi, my name is Dongho Kwak, and I'm a PhD candidate at RIPMO. And today I'm going to be talking about what we mean by music for cells. So as Thomas has mentioned before me, rhythm is one of the important parameters that we are interested in Arduino project. We at RIPMO, especially as an interdisciplinary research center at the musicology department at UIO, have a number of different research areas, and we study how music and sound affects us in many different ways. But also for us, rhythm is something that we consider as a key element in many of our research areas, and that's one of the main common themes that echoes throughout our different areas of interest. And I'm particularly interested in the rhythms and sound of the body that we can find in our body. And we can find these rhythms and sound almost everywhere in our body, but we typically find them where they have a vital function, such as heartbeats and cardiovascular system and respiration, and also in our locomotion. Through the eyes of musicology and music technology, these phenomena, which is comparable to certain musical elements, are indeed quite fascinating. And many of these different parts have their own unique pitch and rhythmic and periodic structure. Although in real life, things are not as straightforward as this simplified illustration that we see on our screen, these basic but different elements are constantly interacting with one another and form some sort of a harmonious state, which is referred to as homeostasis in physiological terms. Beyond what we can see with our naked eye, we find that these vital rhythmic features are driven by what is generally considered as the smallest unit of our body, cells. So as my colleagues have already introduced it, in our Arduino project, we aim to use acoustic and mechano-based stimuli to help making the insulin-producing cells through stem cell differentiation. And through many years of research in the field of mechanobiology specifically, researchers have found that the physical properties of the cells can be understood by analyzing the cellular responses against different types of mechanical probing. And also there's a very complex response system within the cells when external physical forces are applied upon them. And these physical forces play an important role in the life processes at cellular level, such as the fate of stem cells. And although how sound pressure waves, as Thomas mentioned it, that's applied upon the cells have a biological effect is still unclear. There are several reports of examples of yeast cells, for example, vibrating at certain frequency, and how altering the conditions like temperature and drug effects the vibrating frequency changes, and indicating how vibration is vital sign of a different state of the cells. And perhaps sound pressure waves are capable of manipulate the cells in certain way, and perhaps there's a breakthrough to be made. And indeed, last decade or so, there have been efforts by the researchers to understand the effects of sound pressure waves on the cells. And there are several studies that, and they can be largely divided into groups of bacteria and mammal cells, including human cells, and also further into groups of simple and complex sounds. A wide range of frequencies, as you can see within the audible range, was tested, and different outcomes of these studies are quite encouraging and show that cells do display biological responses when sounds were played to them inside the lab. However, we see that there is perhaps a need to turn our attention to signals that we get from the natural human bodies. When more physiologically relevant stimulus is supplied to the cells, we could perhaps expect more natural biological responses of the cells. And also these rhythms and sounds of the human body are perhaps very musical. With this framework in mind, we are studying and investigating the physiological rhythms and sounds of the human body. And also with our music technology and digital audio signal processing and synthesis background, we aim to develop a theoretical and experimental model of controllable sound parameters based on the physiology as cellular stimuli for the Albino project. And answers might lie within ourselves if we listen carefully. Thank you.
Thanks a lot to Albino's research fellows for introducing our project. Now we're going to have a short music performance. Here you will see Albino PhD fellow Dongho Kwak that just spoke. He's performing together with a Ritmo PhD fellow Chichao Lan. And they perform what we call live coding. This is a type of music performance where the music is programmed in real time. So the code that you see on the left is actually what produces the sound that you hear. And they use a language called Glicol, which is developed by Chichao Lan in his PhD. The performance is built around the rhythms found in the human body. So this is connecting to the topic of the Albino project. So enjoy this performance, and we will soon get to the panel discussion. So thank you very much to Xiao Lan and Dong Ho Kwak for performing here for us at this life science event organized by the Abino project. Now we are going to move into the panel discussion, and I'm very happy to be here together with three of my colleagues working in the project. And we are going to have a little discussion on the topic of interdisciplinarity. This is something that we are interested in because we are a highly interdisciplinary project team, and trying to work together is perhaps not so easy, or perhaps is it? We'll hear. when we discuss a little bit about how this is going about. So today we have then Hanne Scholz, who's the leader of the Albino project. She has a doctorate in medical pathophysiology from the University of Oslo and is a senior researcher at Oslo University Hospital and the University of Oslo. She's the vice director and leads a research group at the Center of Excellence that work with hybrid technology organ on a chip at the University of Oslo. And then she's, of course, also then the leader of the Albino Convergence environment. We also have Dag Tyste here, professor of physics at the Njord Center at the University of Oslo. He has worked in interdisciplinary groups with geologists, biologists, chemists, and material scientists for the last 25 years. So it's a lot of interdisciplinary experience there. And now he's mainly interested in the physics of biological processes and has initiated an interdisciplinary student biomaker lab, Lag-Liv-Lab. And also we have here Anne Danielsen, professor of musicology and director of Ritmo Center for Interdisciplinary Studies of Rhythm, Time and Motion, where I'm also working. And she's an expert on popular music and Britain, in particular groove and other things, and is of course also then very into interdisciplinary research. So when it comes to interdisciplinarity, this is something that people often think about. I mean, you can have disciplinary work, which is kind of working within kind of your own discipline or framework. Then you can have different types of multidisciplinary work, cross-disciplinary work, interdisciplinary work, transdisciplinary even. I'm not really sure where Albino is on all of this. We can get back to that. But at least it's a kind of a complex thing to think about. So my questions to the panel to start will kind of be dealing with the interdisciplinarity. But also, when you're watching this stream, feel also free to ask questions to us, to the panel, also about the content of what you heard in the presentations, and we can also try to answer some of those. And if you're not able to answer these questions now while we are here live, we're only running here until 5, then we can also type some answers in the chat afterwards. Okay, so let's get started then. Hanne, leading the Albino project, how on earth did you come up with the idea of putting together such a radically interdisciplinary project?
Yes, thank you. Good question. We heard the talk from the research fellow, and we heard about stem cell differentiation and that diabetes patients need these cells to live, and we all need them, of course. But making them artificial or from a stem cell and tell these stem cells to become insulin-producing cells and be able to work in the body to regulate blood sugar is difficult. And what we have learned since the developing of stem cell technology, since the breakthrough from 2007, is that the cells are, we can push them and we can make them produce insulin, but they are, the differentiation is not efficient, and also they are not mature. So there is still lacking some parts of the differentiation. And what we believe, I think that it's needed something else. And that's why this idea come up. The development of the body is in 3D, is in stimuli from a lot of other things. It's stretching, it's different stimuli that we cannot mimic by just putting in a drug to the cells. So I think that's the key to be able to achieve this goal. So that's why we came up with this interdisciplinary thoughts of working together in such a different field.
Fascinating. And then moving on to you, Dag, a physicist. And then many people think about physics as the hard sciences, kind of dealing with the hard stuff. But then now we're talking more about the softer part of things and biology. Why would a physicist be interested in biology?
Well, there are two main reasons. One is that there is lots of physics in biology. When you get down to the small details, forces and transport, there are lots of physics in there. And it's very exciting to be allowed to be part of the greatest revolution that we've got in natural sciences right now, understanding life. The other thing is that there is actually good physics. There's new physics in there. Yes, we many physicists deal with softer things, but inanimate things. But now there's a whole new field working on active soft things. And the physics of active materials is completely different. And phase transitions we are used to from all the time from Archimedes, saying that there's earth, wind, fire, and water. Those are the four phases. But inside of each of our cells, we can maybe describe some of the complex things going on there as phase transitions. And this is new physics. It's exciting.
Great. And Anne, you're leading a very interdisciplinary research center on its own. And then you're adding on top of that, you're adding this interdisciplinary project. How is that?
It's challenging, but it's really, really fascinating. I remember when Hanne, we met the first time and you described this project to me, I was completely thrilled. And I was also had some problems seeing what are we going to contribute to this enterprise. But then, yeah, I understood why. But I think the working interdisciplinary is challenging. One should not underestimate the challenges linked with working across disciplines. It requires mutual respect, mutual curiosity, and also I think also we need to acknowledge that we come from very different traditions with very different standards, different values, and even different research aims. I think I've experienced through the Albino project that the physicist is interested in explaining things, make things simple, and find the explanation for why things happen. In psychology, which is also a discipline in Ritmo, but in psychology and medicine, I think you're more concerned with whether things work. The aim is, or the goal is to find a treatment that actually makes the situation better for the patient. And we, in music, we are working with, when it comes to sound, for example, we're not so interested in treating sound as, for example, intensity separately from pitch or from rhythm, because music is interesting when all those different aspects are interrelated. So that's also a big difference. But I think we need to respect each other and kind of use the best from the different traditions. But what we've also experienced in Ritmo is that what you think is impossible when you start, actually after some time things develop, people start talking together, there is a dialogue. After some time, it's a very interesting dialogue going on, but it just takes some time. So I think people tend to underestimate how much time you actually need to build a real interdisciplinary research environment.
And now we have been going for about a year in the project, so we are starting to really get to know each other. The research fellows have really gotten into things and collaborate very well, and you heard the presentations from them earlier today. And then when you're thinking about, there are numerous challenges here, of course, but there are also many possibilities. And kind of can hear from you and perhaps start with Hanne about some of what are your ambitions here, and what can we actually really get out of this, and what can also be perhaps the outcomes of this in the end of this type of project?
Yeah, I think that follow up what Anne said, that it's important to be able to get out of your comfort zone, because you cannot stay in the small area where you used to work. You need to go further on, and with that you also open your mind in a new way. You learn new things. You learn to see as you said, the physics thought of the problem with new eyes compared to what you used to do looking at the biology's view. So I think this is that you can get a little bit out of that, and with that I think maybe there will be new development or new discovery that you haven't thought about, that you can dump into because of the interaction among people. And it's also interesting that being able to interact in the way we're building new equipment that can be used also not only for this project, but maybe for others to study details that we normally not have been able to study because we have developed it in this project. So I think this is something that will come up out of this project also, that there will be spin-off for the future. And this is also one of our goals in ABINA, that we want the project to continue also after our convergent environment are come to the end. If we are able to achieve our goal, that need to be seen. But investing time in the beginning, I think then we have the best possible way to get a good outcome. I do think.
But I think we can say that there are always many goals in such a project, because we've got the overarching goal that Hanne said, well, yes, we'd like to make better cells from stem cells. That's the big overarching goal. But of course, on the way, I'd like to understand why things happen, as she says. And I'm sure that you as musicians, you've also got other goals. You know, does our feeling for rhythm start at the cellular level, or is it on the organism level? And it's okay, but this is one of the interesting kind of dances of an interdisciplinary project, that we go with each other's goals, but we also are allowed to follow our own goals. But if we forget the others, then it breaks down.
And then, in your opinion, what has been most fascinating for you so far? I mean, in this interdisciplinary kind of endeavor.
To me, the most radical difference here is working with musicians. Because to start with, I didn't even know whether I believed that sound could really have any effect on cells directly, because I didn't understand the mechanism. And then going as far as thinking about rhythm, that was, far too far away. But now the latest results we've got, we actually seen different rhythms of different cells responding to sound. And, life is so complex that, yes, the complexity of rhythm may absolutely have a place there. And it really takes me time to get used to this idea and trying to understand how can we do this together.
I see now there's activity in the chat here now. So let me pick up on a couple of these questions and put them to the panel here. I think there's one important question here to start with that I guess that you could answer, Hanne, because it's kind of related to the overarching goal here. So Marcy P asks about, so we could put sound waves into the same category, such as physical drug, or kind of use that instead of a physical drug. Is that kind of the overarching goal here?
Oh, not exactly that but we understand that we follow the differentiation part by mimicking the development of the organ in the body. So it's kind of what we mimic is 9 months going on in the human body to develop the pancreas where the insulin producing cells is. And then we want to mimic that in the dish in the laboratory for 3 weeks. And we have learned a lot by the research of what kind of drugs is needed to tell the cells to go from one state to another. But what we believe that this is not the whole thing. So what we will to add in the stimuli, which is learned by mechanical acoustical way of putting energy into the cells, because we believe that there is some stretching going on when you develop as a human being. And as I said, everything is going 3D in the body. So we think that we need to add it. It might be also that we can take away some of the drugs and replace it, so we get a more simpler model, which will be an advantage if you want to develop this for a treatment, of course, or to develop the cells for modeling diabetes and diabetes research. So it will be a combination, but obviously there is not enough what we know today. So we need to improve that, and we think this is the way to improve it.
Great, thanks for that clarification. There's also a question here that I think could go to Anne about the sound, and this is from Soul Rec, asking about the characteristics of the different sounds that we are using now at the moment, and or can potentially use. Do you want to say something about kind of the sonic stimuli we're talking about?
Yeah, so far we have used quite simple sonic stimuli, but we would like to vary the different aspects of sound in a controlled manner. So one thing to think about is, like Thomas mentioned in his presentation, intensity. How loud is the sound, and what do the cells like when it comes to volume? We know also from the literature that if the sound is too loud, it doesn't work. So that's one important aspect. Pitch, of course, is another one, and also whether it might be that one pitch or one note or just one beat is too simple also for the sound, that they actually prefer more complex stimuli. We don't know, but that's also another aspect. And then, of course, coming from the Ritmo, which is, we are interested in rhythm, of course, we are also very, very concerned with testing out whether rhythmic patterns, if that's also an important thing to consider. And of course, there are endless variants of rhythmic patterns, so we will probably go for pretty simple rhythms also. And then we have, because we have also been thinking quite a lot of, as Dong Ho told us about in his presentation, about what kind of rhythms are actually going on in the body. And we have studied with the heartbeat, for example, is not like guk, guk, guk. It's a cha-chung, cha-chung, cha-chung. So it has a certain shape. And also the frequency or the heart rate varies over time. Yeah, so there are many, many different aspects to consider, but we're trying to follow a plan and do it quite systematically. But as I said, yeah, investigating the rhythms of the body is like one interesting place to start.
Just a small comment, because I think that's super interesting, and this is also some complexity for the audience to understand, is that when you have cells outside in the laboratory, they need to be in water and water-based. Because otherwise they dry out and die. Of course, they need nutrition and they need to be fed as we need to eat. So then it's the complexity is also that you need to transfer those signaling and those stimuli into a water-based solution, and also the outcome of what you want to read out, what you have developed from the cells. Everything needs to be very tiny and small and go very, very fast. So this is some of the challenge that we have been discussing during the last year, and then we will continue to develop it. But this is complex, but extremely important, I think.
See that there are several questions coming in here, but unfortunately we are reaching our time limit today. We thought we should do this short and sweet, so we will have to end here, unfortunately, but the Abino project will continue for several more years. So we do encourage you to follow our web pages, where we'll post more information there. Publications will come, also the videos that you have seen today will be posted there. And we will also then be able to follow up on some of the questions and comments that we have here in the chat, also in writing later. But thank you very much for joining us today, and well, see you another time.